I will share my screen. So actually the topic is actually just brief about the application of bioengineering for eco-friendly ground improvement techniques, which is authored by me and my doctorate student, Najam Wani. And I acknowledge his support for the preparation of this paper. And actually the ground improvement, why we need ground improvement, as we see the failure of various structures, retaining walls, embankments, roadways. Therefore, actually the ground improvement techniques are used to prepare the ground for new construction projects for safe and environmentally friendly constructions without any failure. And how to identify the soil deposits? Actually, we categorize them into two deposits. We say stable soil deposits, which are very strong enough, do not need any this treatment for the construction purposes. On the other side, we have marginal soil deposits, which are weak and cannot carry applied load safely and fail, which cannot be used as a construction or a foundation material. Hence, ground improvement techniques are required for safe geotechnical engineering design. Now, some properties about the weak soil deposits, as we see that they have very low shear strength. You can see high compressibility, settlements is more, they can liquefy, there can be high spage losses, there can be foundation instability or acceleration, and higher pressures on retaining walls if not a proper fill material is used, and they need to be modified or treated. Likewise, we have soft clays. Moisture content is almost equal to or more than sometimes liquid limit. Cohesion is very less. SPT value is very small. Wide ratio is too high. Compressibility is also very high. Coefficient consolidation is high, and they need treatment. So major problems are again same: very low bearing capacity, high settlements, rate of settlement is very slow in case of clays. Soiling and cracking problems are there, particularly in case of expensive type of the soils, deep-seated failure, lateral earth flow under the surface leading, etc. And likewise, if we talk about the sands, loose sands particularly, and we can see that they are also problematic and they also need to be modified for the safe construction purposes. Now, coming to the uses of the soil material, actually, we use the soil as a foundation medium. Either we can have, for example, we have this shallow foundation, or we can have a deep foundation. And either we can use it as construction material for structures. All the superstructures are constructed on soil. This we can have a very high-rise building. Likewise, we use it as fill material behind the earth pressure, so retaining walls. If not a proper material is used, then there can be a development of poor water pressure, and hence there can be this retaining wall failure. So what we need is ground improvement technique, wherein we can go for the bearing capacity analysis, test for bearing capacity of soils, and improvement capacity as per the Indian standard codes. Now, what are the criteria actually which will help us for the selection of a ground improvement technique? We need to know the bearing capacity, settlement, seepage, long-term stability, short-term stability, liquefaction, environmental problems, and the most important factors which will be affecting are the purpose of the improvement project, type of the soil at site, its initial properties, likewise area, depth of the soil to be treated, time factor, what is the time factor for the project, and material availability, whether local material is available or it is to be procured from some other place. Then equipment, what are the equipments we have there, skilled labor is or not, feasibility of construction, likewise local experience and local history, environmental factors, and cost effect is above all very important for selection of a ground improvement technique. Likewise, we need to have this soil map, we need to do boring, SPD tests, etc. to define the various soil deposits in different layers, in stratified layers, and so on. Likewise, what we need to know is actually we should have a very dedicated team for the identify the needs of the project and list the solution. We evaluate it properly, then perform the analysis and implement the design. And we need to see whether what we are doing is credible, objectives are obtained, is it reliable, is it reasonable, is accurately done, and what is the current state of, and then whether the relevance is there or not. Likewise, now we are having, there are a lot of ground improvement techniques. We are having traditional ground improvement techniques. For example, maybe initially we may avoid the particle site, we may relocate it. If not possible, then we may design our structure according to the site. We can go for a raw foundation, design very stiff structures, or choose a very flexible construction, and so on. or we can remove if it is a shallow depth and we can replace the unstable soil by a good material if it is locally available, it is under the project cost, or attempt to modify the existing ground, that is by means of applying any ground improvement technique which is most suitable one. So therefore we can have here, if it is a very shallow depth, we can remove it, go to the hard strata, fill it back, or likewise we can go for stabilization techniques. We'll see what are the most important these techniques. Now we categorize them into three categories. One we call as the ground improvement, while we can go for surface compaction, we can go for deep compaction, and we can go for drainage. We can have a this electroosmosis, maybe it is very costly, but it is there for the academic interest. We can go compaction grouting, we can go blasting also, and all these categories are called under ground improvement. Second one we call is a treatment, while we add additives to the this marginal soil. It can be mechanical additive or it can be chemical additive. We have a cement, we have lime, we have fly ash, is a very burning topic and very problematic this industry byproduct which is produced, and we need to use it scientifically, or we can go for dewatering, or we can go freeze thaw and improve the soil accordingly, or vitrification. And this is the techniques underground. This is what we call as a treatment. Now, third one we classify as a ground improvement reinforcement. Likewise, very traditional one. We have a stone column, very interesting. We have soil nails. We have deep soil nailings. We have micropiles, very interesting topic. We have jet growing. Then we have family of what we call as geosynthetics for various purposes, different types, geomembrane, geotextiles, fiber reinforcement is the budding topic now. Most of the people are working on it. Lime columns, weber concrete, mechanically stabilized, very old technique we can use for the modification. But the important one, which I will be just giving an overview today, is the bio engineering or geo-environmental engineering or geochemical engineering technique, in which we can use the this is a technique which includes actually many techniques, and out of them most important one is the vegetative covers, plant roots, microbial induced carbonate precipitation, means we use bacteria in this. The most important techniques which people use are MICP and EICP. So therefore you can use here, we can use the plants, we can just use the roots so that the soil is not eroded away. It is best for the soil stability analysis and erodive soils. So bioengineering is actually a technique for the bank or slope or a stream, this river stream bank stabilization, that incorporation the use of vegetation, engineering structures to increase the slope stability. And stabilization is now emerging as a more cost-effective, aesthetically pleasing, and environmentally acceptable also. Now, the important techniques what I will be covering here is actually the first one is the vegetative cover, in which we can use the plant materials, living or dead, to alleviate the environmental problems such as shallow, rapid landslides, and eroding slopes and stream banks. And in bioengineering system, plants are an important structural components. You can see here figures we can have here. There's a green grass over here, which can also avoid the erosion of the soil. Now, microbial, the second one is the microbial induced carbonate precipitation, where carbonate precipitation is actually produced here by means of bacteria. So this is the geotechnology and innovative one, also eco-friendly technique that uses the microbes to improve and strengthen the weak or marginal soils. Biomediated ground improvement techniques have gained a lot of attention among the researchers since the past decade. Now, third one is the EICP, which we call as enzyme-induced carbonate precipitation, which source goes back to the plants also, has the potential to be applied as a biocementation and bioremediation solution in many environmental, construction, and geotechnical civil engineering problems. So therefore, among various bioengineering techniques, effective use of natural grasses has turned out to be the best available ground improvement technique for the treatment of the erosion and unstable slopes. We can see here, we can have a slip failure here, infiltration. There can be a problem because of rainfall. And to avoid here the erosion and the slope failure, we can go for the plantation, which can actually hold the soils together and give you an environmentally good aesthetic looking also. As reported by the Freer in 1991, bioengineering system plants are important structural components. Likewise, these are some various methods which we can have different biotechnical engineering techniques. Where the fashion where you can put dig a hole and put this plant into the soil and fill with the soil. These are the various techniques people follow for the safety of the slopes and the erosion control. Likewise, it has been reported, various researchers have reported that the institute tests were done wherein the soils were tested without soil roots and with soil roots. And you can see the extreme left figure wherein there is a failure without soil roots. You can see here that the failure plan without is very smooth and there is no encourage or bonding. Likewise, on the other hand, B or C, whether their roots are mixed with the soil, is very massive system in the extreme right side, and it is very difficult to dislodge the soil particles. And hence, you can see that the roots play a very important role in the stabilization of the those loose soils or weak soils. Likewise, we can see the pull-out resistance has been reported by the various researchers, and we can see the here the left one here without root has been tested. You can see failure is very smooth. Exactly. Whereas on the other side, you can have the typical displacement, and without this is shown here, which shows that the presence of branch root influences the pull-out behavior over here, where you can have massive these roots and they cannot give failure. They can have higher resistance and higher strength. So without branching, the roots fail by simple pull-out. A distinct peak, we can say here, very distinct rigid one is shown here. Whereas in case of roots, where there is a progressive failure actually, you can't have a distinct abrupt failure. And these are some examples of the field work which have been reported by the various researchers. We can also go to the failure slope. We can identify the area which is unstable. Then we can introduce the construction, and finally we can get the complete bioengineering research site, which is very aesthetically also good, environmentally also very acceptable. And then we can have a this vegetative wire mesh mechanically stabilized earth retaining wall. And here we can see the bamboo. These can be micropiles, and once they are all, you can fill the soil over here. Then after that, we can have very green environmental. And these conditions here are very aesthetic and very useful. Likewise, some laboratory tests have been done, and it has been seen that, of course, there is a increase in the shear strength. So at 15% water content cohesion in root reinforced soil increased with a range of 21 to 29 kPa, varying root length is given here. It is reported by various researchers. So actually, what is the mechanism? The root architecture actually plays a very important role in the root soil interaction mechanism. Different idealized root structures are shown in the figure. We can have a shallow one, which is parallel to the earth surface, which is known as a H type. Then we have deep branch root type. We have strong vertical and horizontal, very deep one. Then strong vertical oriented and profuse. There are many more, but I have just listed few one over here. And it has been reported that VH type, H type, and V type means H type here A and C and D have been found to be very effective in slope stabilization, wind resistance, and increasing the shear strength of the soil. Studies are also shown that a tensile strength about 60 MPa is attributed to the root of different vegetations, and vetiver root length varies between 1 to 3 meters, which makes them effective for reinforcing the soil against natural effects of the heavy rainfall and wind. So accordingly, if we see that the shear strength, actually the Moore-College equation is modified for the biostabilized soil, which is expressed in one more component because of the cohesion, because of the this is a CR, means because the root is the apparent cohesion, just like what we have apparent cohesion in unsaturated soil. It is something like that, which initially gives us high strength. The in situ tensile pull-out resistance is given by TF into, this is the stress into the root area. So overview of the different reactions and outcomes which eventually lead to alter the engineering property is shown in the figure over here. We see that because of the shear strength and there is a compaction of this compacted soil stuff, we have less permeability, we have higher stiffness, compressibility or settlement is reduced, shear strength is increased. So we can have the biomineralization, biofilms, and biogas. These are the steps which has been reported by the author given at the bottom of this slide. Likewise, various advantages using this technique. First one is a technical advantage, which includes protection against surface erosion, increased slope stability by root reinforcement and draining of the soil, protection against rockfall. Then ecological advantages, which include actually regulation of temperature, humidity, close to the surface, promoting growth, improvement of soil water regime via inception, evaporation, and so on. Then soil improvement and topsoil formation, improvement of the land, and provision of the habitats. Then the third important one is economic advantage, which includes actually reduction of construction, maintenance cost, creation areas for agricultural land and recreational use. And overall, the last and not the least one is the aesthetic, which includes structure fit into the landscape. Landscape is more appealing. So these advantages make biotechnical technique a worthwhile consideration in stream and bank rehabilitation. The following chapters, these advantages are discussed in the great detail. So this is how the environmentally we have aesthetic look over here and green environment. However, this is technique, but there are challenges also, because we may have accessibility problems. We may not access the site. There can be a lack of adequate data for the design of protection or restoration of the work. Vulnerability of bioengineering projects immediately after the installation due to plant mortality or other factors, or previously applied ineffective protection measures may be there, which we may not be we can't be rectified. Need of participation from local residents. Local history is the important, actually, how the area behaves, how the previous structures were there. The limited availability of plant also can adapt the site conditions, limit time for installation, availability of skilled laborers, high maintenance requirements, or also other factors which add to the challenges of this for the implementation of this soil bioengineering projects. So thus, poor selection of the species or the generation technique can lead to the failure of bioengineering project as well. One has to be very careful. Now, then the second technique which is being used is MICP, actually, which is nothing, which is the urea hydrolysis utilizing bacteria as a source enzyme for soil cementation. It was first discussed by the Whifflin in 2004 and later called as microbial induced carbonate precipitation. Various studies have examined the potential of this technique in addressing many challenges in granular soils such as erosion resistance, slope stability, underseepage levees, and bearing capacity of shallow foundations. This is an innovative, eco-friendly technique that uses the microbes or bacteria to improve and strengthen the weak soils. Biocementation, bioclogging, achieved through microbe action with the help of accelerated supply of nutrients and cementation solutions work together in this technique. Flocculation, densification of soil occurs due to enhanced bonding. Because of bonding, shear strength is increased, compressibility is reduced because of particle to particle contact. Then this technique is a process in which the calcite formation is achieved as a result of the active metabolism of bacteria, which generates a favorable microenvironment for the precipitation. So actually, this technique requires the knowledge from different fields, including biotechnology, geotechnics, civil engineering, material engineering, and nanotechnology and environmental engineering. This is the schematic diagram, how it works. We have a on this untreated soil over here, loose soil, for example, untreated one. Then before MICP, then after that, when the COC crystals are produced and the biochemical action takes place, then these loose particles are actually bound together within the soil mass, and here it is shown an illustrated figure 1, and hence these particles are now contact to contact, and hence compressibility is reduced and strength is increased. And accordingly, the various researchers have also reported the scanning electron, which actually verifies that there is a contact to contact program. You can see here various these calcites formation, fine soils, poor whites over here. This is biostabilization of fine silty sand. And this is the schematic for the self-healing of biocement. You can have this dormant bacteria actually, which is dead. Then when it is activated, because of some crack or whatever, the insertion of air here, it gets activated. and then because of the MICP process starts, and hence the cracks are filled by this precipitation, which bonds the particles, soil particles together, and hence the compressibility is reduced and strength is increased. Likewise, there are some other these scanning electron images, which actually of sands provide visual perception of the distribution, size, packing, and density of the precipitated crystals in these two figures. Now, in the third technique, which is actually EICP, which employs the urea enzyme to precipitate from an aqueous solution of calcium chloride and urea to fill the soil pores, and which actually increase the dilatancy, reduce the permeability, consolidation parameters, and cement soil particles increasing the shear strength. So actually, this EICP is a very innovative technique that involves the calcium carbonate precipitation through the hydrolysis process of the into the ammonia ions and the carbonate ions catalyzed by the urea enzyme source from the plant. Actually, carbonation is the chemical process of carbon dioxide to give the carbonates and the bicarbonate. This is the process. You have soil, and because of the biochemical process, the precipitation is there, which goes in between these soils and binds them together. Hence, this is the chemical their equation given below. This because of hydrolysis, then we have treated soil sample by means of EICP technique. The mechanism involves here actually the process of urea analysis, where urea is first hydrolyzed into ammonia and carbonate catalyzed by the enzymes. The chemical reaction involved are described in these equations over here. Chemical equation actually not too good in these equations, more related to chemistry type. And these are the SEM images, which shows these particles are being coming together and being compacted here, while these are being reduced, while sand grains are coming closer and closer. There is a precipitation in between them. Here we can see this clearly. And this is reported by the Zimmer in 2020. Now, something what we have done,
Professor Mir,
done in our lab.
Professor Mir, can we conclude in 2 minutes, please?
Sir, 2 minutes, time is gone. Oh, sorry, sir. Actually, what we have done in IGC Srinagar with my drafted student, this is the layout of our research work. I cannot discuss all here. And we have done some fabrication also. I will show directly the fabrication of the UCS molds, which are shown over here, fabricated molds for the treated soil samples. And the methodology is that we have chosen the bacteria, then some sample tests have been done so that we can choose the weakest soil, and then we add our program as per optical density and the mortality preparation, so that we can do these are the soil sample site where we have collected the soil samples from 3 the collections about near the Jhelum River, and these are some site photos. We have taken the soil. This is the collection of soil samples, disturbed and undisturbed soil samples. These are the basic properties of the soil, untreated soil samples, and the soil one is actually the weakest compared to the other two. This soil sample site has been taken for the improvement, and accordingly, these are the basic properties continuing. Then the bacteria we have chosen, these are the 4 types. Actually, it is difficult to even pronounce them, but my student has worked very hard. These 4 microbes have been chosen for our research work. All these microbes are either non-pathogenic, cause minimum disturbance to other living creatures, since they are all present in the environmental and hence are part of our ecosystem. And what we have done actually, how we have prepared here, different optical density values, 0.4 to 0.2, equivalent to 2 into 10⁷ cells per milliliter were used for the respective, and this cementation solutions was prepared. The basis was chosen through extensive literature survey. First set was prepared in the laboratory, which included all basic in situ test and remolded tests. Second test was on MICP. The bioaugmentation approach was used for the research, in which soil was mixed with this cultured these microbes, bacteria in our laboratory. And UCS test, shear strength density test, California bearing and paleo tests were conducted. So these are actually how this
last 1 minute. Can we conclude in 1 minute?
Sorry, this is the bacteria cultivation, and this is the final solution. These are the single colony, and we are receiving microscope images. This is our full laboratory test where we have this where samples are being treated, and These are some basic tests of treated soils. I have put some very selected one of, we can see that we can improve the soil samples. You can see again here, strength is increased. Likewise, shear density test, we see that shear strength is increased. Is a comparison, and this is the soil after testing. We see the failure plane has been recorded over here, and the CBR test also shows the increment compared to the untreated soil. Then this is the comparison of various those bacteria at different moralities and cementitious moral solutions. And some SEM have been taken. We have taken the soil sample along the failure plane, and this is the SEM this which also verifies our test results. You can see over here, precipitation CSO3 formation is over here. You can see interlocking is being shown over here. Cementation clogging is shown here. And these are some consolidated data against different moralities and the curing periods. We have gone up to 28 days period of curing. And this is the UCS test, MICP, and you can see the how much strength. There's a drastic increase in the shear strength, and permeability has also decreased. Various researchers have also has been validated our results, and it is validated that because of this permeability is decreased, compressibility is decreased, and these are the conclusion. Actually, the in short conclusion is that this is a very innovative techniques, and we can use this. But however, some challenges are also there which need to be taken before the selection of the proper site selection, equipment, and these are some references. And I thank you for attention. And any questions can be mailed at our email so that we can give a right answer for the right question. Thank you, sir, very much. I hope.
Thank you. Thank you, Professor Mir, for a wonderful presentation. I am sure all the delegates attending this session will be greatly benefited.
Thank you, sir.
Friends, let me tell you, this bioremediation I recently did very successfully on New Shimla Airport.
Thank you. Yes,
yeah. So it's a very, very nice presentation, Professor Mir. Well, friends.